Hello, welcome to my channel once again. My name is Maxwell Asiedu, a PhD student in plant physiology. On this channel, we speak mainly about applications of bioinformatic tools in plant biology research. So in our last video, we spoke briefly about a general overview of GWAS, which is genome-wide association studies. We talked about the concept behind GWAS, the input files you would need to perform your GWAS analysis, and how you may want to interpret your GWAS results. So if you are new to this channel and want to see that video, kindly have a look at the top right corner. I've staged the video there. Please go have a look and come back to this video. So today we are talking about genotypic data filtering. So before you get a good quality DNA or SNPs for your research, for your GWAS, you would need to do various quality control analysis. And one of them is minor allele filtering. So the minor alleles are the less common alleles across your population. So for instance, those alleles that are in less than 5% or 1% of your population. Those are the minor alleles that you want to filter. So I've tried to come up with a very simplistic overview of what minor alleles are. So let's say you have alleles A, B in 200 genotypes. You find that allele A is in 198 out of 200, and allele B is in just 2 out of 200 of your population. So this tells you that allele B is a minor allele in the population. And so these are the minor alleles you want to filter or remove from your SNP data set because they are less informative, and your GWAS models may not be able to, if they are significantly associated with your phenotype, the GWAS model may not be able to detect these SNPs. All right, so you want to filter these SNPs from your data. And then secondly, there are some SNPs that have a lot of missing genotypes, so you want to filter those ones out as well. So usually you want to filter those ones with more than 10 or 20% missingness. And then lastly, you want to do the data imputation of the unknown genotypes in the panel. So some SNPs have genotypes that are unknown, so you want to impute those unknown genotypes, and this will help you get a very high quality SNPs for your GWAS analysis. And so with this done, what we are going to do next is I'm going to walk you through how to get a sample data set, how to filter those data sets using TASSEL, and then lastly how you are going to impute the unknown genotypes using Beagle. All right. Right, so what we are going to do first is to download our genotypic data. And even before we download our genotypic data, you make sure you create a folder on your desktop. Call it, so I've called my GWAS tutorial. So you can create any folder create a folder on your desktop and call it whichever name that you feel is okay. So I've called my GWAS tutorial. Now let's go to the browser, and then we will just type in GAPIT. All right, so you type in GAPIT, GAPIT, and then you go to this Maze Genetics website, this link, the first link here. So you click on the first link, it will direct us to this website here. So the GAPIT is the Genome Association Prediction Integrated Tool. So what we are interested on here is you scroll down till you find this source code and demonstration data. So you look for demonstration data sets, click on the link, and it will automatically download the input files for us. So what you just have to do is to copy this file. The downloaded file should be in your downloads, so you copy that, and then you just simply paste it or place it into the folder we created on our desktop. So you're going to have a zipped file just like this. Place it in your GWAS tutorial or the folder you created on your desktop. All right. So we have to unzip it, open it up. So we just open the file. And now what we are going to do next is to look for the genotypic data as well as the phenotypic data. All right, so before that, you want to create another folder within the GWAS tutorial or the folder you created and call it, so I'm going to call my input files. All right, so we go back to the GWAS tutorial data, the data we just placed inside the GWAS tutorial. So here you would have a lot of files, but what we are after is the genotypic data. So you have the genotypic data or SNPs for each of the chromosomes. So this is a maize sample data, so it has 10 chromosomes and they have the SNPs for each of the chromosomes. But what we are after is the data set with all the chromosomes together. All right, so you don't want the chromosomes separately, we want the file with all the SNPs for all the chromosomes. So that is this. So you copied the file, so that's the mdp genotype test.hmp.txt. all right, so that's what we're after. So you copied that together with the phenotypic data. So the phenotypic data is called the mdp underscore traits.txt. So you copy these two files, and then you place them into the input files folder you just created. Great. So now we have our genotypic and then the phenotypic sample data sets. So what we are going to do next is to filter the genotypic data for the minor alleles and the missingness. All right. So to filter our data, we are going to use TASSEL as a tool for the filtering. So if you don't have TASSEL already installed, you can simply go back to the website where we just downloaded the input files. Go to Bioinformatics tab, and then you see the TASSEL here. You just click on the link, the TASSEL link, and it will bring you to this website. So on here you find 4 links. So depending on the operating system that you are using, so I am using Mac, so I would have clicked on the first link here to download the TASSEL file. So if you're using Windows 64-bit or 32-bit or Unix, you, depending on your operating system, you just click on the link and then you download the file. So when the TASSEL, the file is downloaded, you just click on the link and then follow the prompt to install on your computer. So I already have the TASSEL uploaded or installed on my computer, so I do not need to install it again. Okay, so what we are going to do next is just open the TASSEL. Great. So we have TASSEL open now. Now we are going to load the genotypic data into TASSEL to filter the minor alleles. So what we are going to do is go to File, Open As. So our genotypic data is in a HapMap format. All right, so you choose HapMap here, then you sort positions, then OK. Then you make sure you are inside the input files where your genotypic data was placed. So we click on the genotypic data here, then you open it. Excellent. So now we have, this is how the genotypic data or the SNPs looks like inside of TASSEL. So the first column on the left here are the taxa names or the genotypes, the names of the samples. So we have 281 samples here on the left, and on the first row here are the SNPs. So there are 3093 SNPs. So what we are going to do is to first filter the minor alleles. To filter the minor alleles, you find a lot of tabs here. So we go to filter, then you have options here: filter genotype sites, filter genotype table, taxa, traits, etc. But we are interested in the first button here, so we just click on that. It will pop up a new tab here. Now on here we have 281 samples, all right, and we want to filter samples that have more than 20% of missing genotypes. Okay, so out of 281 samples, if you want to filter more than 20% genotypes that have more than 20% of the missing genotypes, so 20% of 281 should be somewhere around 56. So you take 56 out of 281, and that should be about 224. I may be wrong, but that should be it. And then the next one is the site minor allele frequency. So this is the most important part here we're going to use. All right, so I want to filter alleles that are in less than 5% of the population, so that will be 0.05. That is it. So it's very simple. You leave the rest of the options to default. All right, so once this is done, you just click OK, and now you realized in the unfiltered genotype data we had 3093 samples or SNPs, and after filtering we are left with 2559 samples. It means the rest are minor alleles that were less than 5%. So yes, we are good to go. Now if you take a good look at the SNPs here, you find that there are a lot of N's, and the N's are unknown SNPs, as you can see here under the. So if you go to the nucleotide codes under the derived IUPAC, you find that N represents unknown SNPs or alleles. So you want to impute these unknown genotypes, all right, and there are several ways we can impute it. You can impute it within TASSEL here, but I prefer to use Beagle. So once this is done, what we are going to do next is that I'm going to show you how you can use Beagle to impute the unknown genotypes. All right, and Beagle accepts files in a VCF format. All right, so one very good advantage of TASSEL is that you are able to convert the genotype format into several other formats that may be useful for other downstream analysis, like the imputation with Beagle we are just going to do. So what we are going to do next is to save the filtered genotype data as a VCF format. All right, so to do that we go to File, Save As, and then you make sure you are in the input files we created. So let's call this test. All right, and then you click Save, and then the format is in HapMap, so you just choose VCF, and then you click OK. It's very simple, straightforward. So what we do next is to check if the file is in the input files, and you can see we have our test file. So this is our filtered file that is ready for the Beagle analysis. So our next step is to do the imputation using Beagle. All right. Now for our data imputation with Beagle, what you're going to do is download a Beagle jar file. All right, so Beagle is a Java tool, so we're going to download the jar file. So what you want to do is go straight to your browser and search for Beagle. So if you just search Beagle, you're going to have a lot of irrelevant articles. So you want to narrow it down to the file, so, or the website, actual website. So you can do, say, Beagle genotype imputation. Beagle genotype imputation. Then you search for it. The first link you find, so you just look for this website, Bioinformatics Home, Beagle, and then you click on the link. Then you move straight down, you scroll down until you find the jar file, and you can see the source here under download and documentation. You find that you have the jar file here, so you just click on this link and it will automatically download your jar file. So what you want to do next is just copy this jar file in your downloads. So you copy the jar file from your downloads, then you just place it in your input files folder in the GWAS tutorial. You just place it here, just like this. Good. So now to impute our genotypic data, which is the test.vcf, you want to head straight to your terminal or your command line. So you will go to the terminal. There's the command line tool may be different in Windows, but on Mac it's just the terminal. And what we are going to do is, you want to make sure you are in your terminal, you want to make sure you are inside. Let me enlarge this a bit. So you want to make sure you are inside your input files to operate the terminal. So we just go to inside our terminal, inside our input files. So we first go to our desktop. So you just type command desktop, and our file is the GWAS tutorial on our desktop. So GWAS tutorial. And the files are inside the input files under the GWAS tutorial. So we go one step further down, input files. Excellent. So we are inside the input files now. All right, so now to impute the genotypic data, you want to enter these commands for the Beagle to impute our data. So the command line is Java. So the next code is just to tell the Java, it's just to provide a space for the Java. So x4g, so you can do 8g or 16g depending on the space on your computer. So I'll just do 4g, that should be enough. And then jar, you call the jar. So this is calling the jar file. So that's Beagle. So you just put the name of the jar file that you downloaded. So that's Beagle.24. You can rename the Java file as you want, but the most important thing is just put the name of the file. 0.3e8.jar. Excellent. And then you now call the input file. So our input file is test.vcf. And next you tell the Java how to output your file, or the name to call the output file. So that's, let's call it test.1. All right, so, and then you ask the Java to impute, or you ask the Beagle to impute. So impute is equal to true, and you just enter. So it's depending on the size of your input files. If you have a very big size, it's going to take a while, but these are just 3,000, about 2,000 snips, so it should take a few seconds. And then we have our file, our genotypic data imputed. So the output will be a zip file. So what you will do next is to just open the file. All right, so to open the file, open the zip file, you can just open it using, what you do is to put gun zip, and then you put the name of the output file. So that's test1.vcf.gz, and that will unzip your file. So when you go to your input file, you find that you have the test1.vcf. Okay, so now we have imputed our file using Beagle. So to confirm if the imputation worked, we will go back to TASSEL, and then we will load our file. We will load our imputed file. So open as VCF. All right. Sort position, keep dev, and then we open it. So that will be test1.vcf, isn't it? We open the file, and as you can see clearly, when you compare this to our previous file, which is the mdp genotype.filter, so the file we filtered and then converted into VCF for the imputation, we go back to that, you find that there are a lot of n's, which are unknown genotypes or alleles. But when you compare the test one, you find that there are no n's or unknown genotypes. We have imputed all the unknown genotypes, and this should be very good for us. So this should be a very good quality SNP data set for our GWAS analysis. So what you want to do, what we are going to do next is to perform our GWAS analysis. But even prior to that, there are several other analysis that we may have to do before we do our genetic analysis, our GWAS analysis, such as population structure analysis. So, but then what you want to do now is you may want to save your file as a HapMap file, because GAPIT accepts a HapMap. So you want to save it as a HapMap file. And yes, so we are almost there to perform our GWAS analysis, but there are other quality control analysis, a couple of them that we may have to perform before our GWAS analysis. So our next video, our next episode will be, or our next tutorial will be population structure analysis. So yeah, thank you very much for this, for staying with me. Now, if you have any question, or if you have any doubts, or anything that you wish I'll explain in detail, please go to the comment section and then just leave your questions there. I will try as much as possible to address all those questions in our next tutorial. If you find this videos educative, please subscribe to this channel. I will try as much as possible to create a lot of these videos, and depending on the questions on your mind or the questions you put in the comment section, I'll try to address all of that in our next sections. Thank you very much, and see you soon.